So this afternoon, I chose to talk about complement, particularly because of its importance in medicine, but also because I realized that on your schedule, complement wasn't included. And yet it's very important that you understand exactly the role the complement system plays in both the innate and the adaptive immune response. Many patients normally come in and they have defects in this complement system, and it really affects someone's response to pathogen. So I hope that by the end of this lecture, you're able to tell the three types of complement system activation. You're able to describe how each of these systems is activated up to the point when we have something called the membrane attack complex. You're able to tell the role of complement, the functioning of complement in the immune system, and you're also able to discuss how complement is regulated. Because as you will find, if the body fails to regulate complement, so many things can happen. A patient can actually come in and they have edema because the body is unable to regulate how complement is being activated. And of course, finally, you must be able to tell which diseases can occur if someone has either deficiency or an excess of the complement proteins. So, the complement system, like the word really is, it only complements the abilities of our antibodies and the phagocytic cells to clear pathogens from an organism. So it means that you must have another way of dealing with an organism, and that this complement system only comes in to add onto what already exists. So whereas the complement system is said to be both innate and adaptive, it's mostly innate in nature. And as you will realize, it really doesn't adapt to a given infection, apart from the way that it's activated. But otherwise, from the point of activation up to the point of function, the complement system seems to act in the same way at each and every given organism. So this system consists of quite a number of small proteins, and these proteins are found in blood, and they are generally made from the liver. But also some of these are made from macrophages and monocytes, and they are usually found as inactive precursors. So the moment you start to activate complement, you end up with a cascade of events which activates these proteins. But initially in blood, the complement proteins are found as inactive precursors. And that's important because the moment you're unable to keep them in their inactive form, then you end up with what we call complement-mediated lysis of cells. So significant amounts of these complements usually, like I said, come from the liver. That's why patients who have chronic liver disease. But also, like I said, complements can come in from tissue macrophages in your urogenital and the gastrointestinal system. And I guess you can imagine why it's the urogenital and gastrointestinal system, because these are the areas where normally organisms enter into the body. So when one of these proteins is stimulated, you end up with a cascade of events that involves proteases acting on specific proteins to release cytokines, and then this ends up initiating one amplifying a cascade, which causes further cleavage. And like I'll be showing you, these cascades are so important because failure of one of these cascades means that you cannot end up getting activation of the complement. But usually, the end product of this activation is amplification of the response. and production of something called a membrane attack complex, which kills the target. This target might be a fungus, it might be a microbacterium, it might be a bacteria, it might be anything. And of course, like we shall see, the moment you're unable to control complement, then your own antigens become a target for this complement. So the complement system has over 30 proteins, and these proteins, like I said, they're usually in blood, so they can also be found in serosal areas, and they also cell membrane receptors. And they account for about 5% of our globulin fraction of blood serum. And they usually serve as opsonins. I'm sure you remember what an opsonin is, because opsonins just really help to increase palatability of an agent. So these complements end up assisting in the production of opsins that help in improving palatability of an organism. So they help phagocytes to function better. Now, when we talk about the activation of complements, we are looking at 3 different pathways through which this complement system is activated. We have the classical complement pathway, we have the alternative complement pathway, and the lectin pathway. Whereas the end product of these pathways is always production of the membrane attack complex. In essence, the activation is different. And we shall be seeing where these differences occur. But by and large, the classical complement pathway requires presence of an already existing antibody, which means that you should have been exposed to an agent produced either IgM or IgG before classical complement pathway can be activated. Whereas the alternative pathway and the lectin pathways are purely innate. The presence of an antigen is able to activate the alternative and the lectin pathway. So that's a very, very specific difference between the activation of these two. Okay, so classical requires antibodies to be activated, whereas the alternative and the lectin do not require antibodies. You only have to have presence of an antigen, and then you activate these pathways. So the lectin and the alternative pathways are therefore more important the first time we get into contact with an organism. And because of this, their activation tends to be slower, whereas the classical one is very important if we are getting re-exposed to an organism where we already have existing antibodies. I'm sure you have already studied about antibodies and antigens. You've done the innate system, you've done the adaptive system. So I guess what I'm speaking of is known to all of us. So the lectin and the alternate pathways are purely innate in nature, whereas the complement is not necessarily innate in nature because it requires antibodies. So generating variants of a C3 convertase, as we shall see, the C3 complement pathway is different. So we have to literally be able to tell that what I'm looking at is a C3 convertase for a classical pathway or for an alternate pathway. Now, the complement proteins, whereas they are innate in nature, they are able to recognize and bind to non-self pathogens. And this is very important. As a complement is innate in nature, it's non-specific. Usually, the body is not damaged. It's because these proteins are able to recognize pathogen-associated molecular patterns, which are normally found on only pathogens. And then they bind, okay? And at the same time, if if you look at how antibodies act, antibodies also tend to have specific sites where they bind. So they will not bind necessarily on our own antigens, but they will bind to the antigen that is foreign. Okay. So complement is able to detect the non-self enemies, more specifically because it detects the pathogen-associated molecular patterns, which are specific to only the organism of interest. But at the same time, like I will be telling you later, regulation requires presence of particular clusters of differentiation. And these are found on only human cells and not on the pathogen cells. So if you lack some of these clusters of differentiation, then complement will not necessarily be able to tell the difference between human and the antigen. So what you're looking at there, the classical pathway and then the alternate pathway. I will be taking you slowly through each of these activations so that we are able to understand the differences between the classical pathway and the alternate pathway when it comes to activation. So if we look at the classical pathway, so we shall start off with the classical pathway. Now, the classical pathway, like I said, usually requires presence of an immunoglobulin, either immunoglobulin M or immunoglobulin G. M is for acute infections, G is for chronic infections. And this is usually triggered by activation of a C1 complex. So this C1 complex is composed of one molecule of C1q, two molecules of C1r, and two molecules of C1s. Now, what happens is that in the presence of an antibody, C1q will bind to this antibody, either IgM or IgG. And once this binding occurs, then you have a change in this C1q molecule, which leads to activation of C1r. So C1r is a serine protease. So for it to be activated, you need to get an antibody, either IgM or IgG, binding to a C1q. Once that has occurred, then you activate two molecules of C1r. Now, the moment C1r is activated, it will cleave C1s, meaning it also activates it. So you end up with an activated C1s, which then starts as our precursor. Okay, so the C1s will now cleave C2 and C4 by cutting them into half. Okay, so these complement proteins are usually named Cs, C for complement. So we have a C1, a C2, a C3, a C4, a C5, C6, C7, C8, and a C9. So the C1r that I've just told you about, the C1 complex, that one has Q, R, and S. But the R and S are the serine proteases, which when activated, they cleave C2 and C4. So C2 is split into C2A and C2B, and also C4 is split into C4A and C4B. Now, this C4B, C2B and C4B will bind on the surface of an antigen. Like I said, this antigen might be a bacteria, might be a virus, might be a fungus. And the C2A and the C4A will diffuse away. Now, at the same time, C4 will also bind with C2B, so that we form a complex called the C4B2B. This complex is what is referred to as the classical pathway C3 convertase. Okay? And it's called the C3 convertase because it is what then starts to activate, okay, or cleaves C3. So C3 is cleaved by C4B to B into C3A and C3B. C3A does not bind to any other cell. So it literally binds usually on mast cells where it can activate anaphylaxis. Okay, so you end up activating histamine. And then, but it's also important as a chemotactic factor. Whereas C3B will go and join C4B and 2B. But before it does that, it has to first literally cover the entire cell to act as an opsonin. So C3B is an opsonin, but at the same time, it also combines with C4B to form C4B 2B 3B, also called the C5 convertase. Okay, now we've seen the role of C1. We've seen C2 being split into C2A and C2B, and we've said that A, so usually anything which has an A tends to diffuse away. Those which don't diffuse, like C3A, C5A, act on mast cells to release histamine. But at the same time, they're also very important because they attract other cells to join into this fight of destroying an organism. Okay? And then we've said that the moment we formed a C4B 2B, this is what we call the classical C3 convertase. This is what splits C3 into C3A and C3B. C3A will act on mast cells, whereas C3B will coat cells as an opsonin. But at the same time, also some of the C3B will bind with C4B2B to form C4B2B 3B, which is the C5 convertase. This is what I'm actually trying to say. So there's something called the C3 activation complex. We have a C4B, a C2B and C4B, which we said come from the splitting of C2 and C4. Now, together, C2B and 4B will complex and they form a C3 activation complex. This complex, its purpose is to cleave C3 into C3A and C3B, like you can see. So we start off with this C2B 4B, which you said is our classical C3 convertase. It's here, it splits the C3 into C3B and C3A. And we said this C3A will bind on mast cells, whereas C3B will bind on the pathogenic cells. When a cell is totally covered, the remaining C3B will bind on the C4B2B to form the C4B2B 3B, which is a C5 complex. So like you can see, many C3B molecules are produced by the C3 activation complex because you're literally splitting a lot of C3 to C3A and C3B, okay? So the C3B will bind to and coat the surface of bacteria, but also can be a fungi or a virus. So this C3B, in so doing, so in other words, in binding to this, it is acting as an opsonin. And this increases the capacity of a phagocyte by over 1,000 times. So if you have an opsonized bacteria, the chances of it being phagocytosed are going to be increased 100-fold. That's what it means. Okay? And yet we said also that C3A, okay, and the C5A, these ones, will also bind on mast cells. And when they bind, they end up activating release of these granules that are containing histamine. And that is why they are very important in allergies. So C3 and C5A are very, very important in in allergic reactions. So C3A will increase the inflammation by binding to mast cells, releasing histamine, but it also attracts the other phagocytes, the neutrophils, the macrophages, to the site where there is inflammation. So how do we build C5? So C5 is built by the fact that eventually it binds onto the surface of the cell, and eventually the cell gets saturated. So the remaining C3 complex is made of C2B, C4B, and C3B. And this will cleave C5 into C5A and C5B. C5A has similar functions like C3A. In other words, it attracts cells, but it also binds onto mast cells. On the other hand, C5B, okay, once you produce C5B, then it also starts to coat the surface of a bacteria, and it is the precursor of the production of C5B, 6, 7, 8, and later 9, which polymerizes to form a membrane attack complex. This is what I'm saying. The C5A will disperse and increase inflammation, and also the other will attract. Whereas for the sake of C5B, some of it, like we said, is an option, so it will bind to the cell, and when the cell is saturated, the remainder will now bind to C6. The binding of C6 activates binding of C7, binding of C8, and later there is polymerization of C9. The moment C9 polymerizes, we end up forming a circular complex called the membrane attack complex. So this is the complex which inserts into the cell is going to die. Okay, so this membrane attack we end up with all these intracellular components rushing out of the cell, water rushes out, and the cell will die. Okay, so that's the way. We start off with a C1R producing R and S, which are sign proteases. These will cleave C4 and C2 into C2A and C2B. The A's will diffuse away, whereas the B's will bind to form the C4B, 2B, which is the C3 convertase. This C3 convertase will act on C3, will split it into C3B and C3A. C3A acts as acts on the mast cells and also attract cells, whereas C3B acts as an opsonin, so it will bind on the surface of a cell, and when a cell is saturated, the remainder will bind with C2B4B to form the C5 convertase, which will then split C5 into C5A and C5B. C5A will bind on mast cells, whereas C5B will bind to cells and also bind to C6, to C7, to C8, and then C9. The moment C9 is bound, you end up polymerization, which produces a membrane attack complex. It is this map or mark which causes channels into the target cell and ending up damaging the cytoplasm, which causes water to rush out of this cell. What about the activation of the alternate or the alternative pathway. This is a bit different, okay? So the alternative pathway is part of the non-specific defenses. So like we've seen, the classical pathway requires presence of an immunoglobulin, either IgM or IgG. This is not the case for the alternative pathway. This one usually only requires presence of a cell surface. This might be a bacteria, it might be a fungus, it might be a virus. And the moment you have any of those antigens, this will initiate binding of C3. And then a factor called factor B. Like you can imagine, if you do not have an antibody pre-existing, then you end up with a slower response. So the alternative pathway is much slower than the classical pathway, okay? So this is what happens. So we have spontaneous hydrolysis that occurs, which produces C3A and C3B. So in the presence of an antigen, this C3B will bind to the antigen, okay? And once it has bound, we then have factor B coming in, and then this factor B is going to be split into BA and BB, so that we have C3B, BB, and then you will have propagandin that comes to stabilize this complex, okay? So you have the C3 contains this unstable, I've already talked about this spontaneous hydrolysis, which produces C3B and C3A. So in the presence of a foreign substance, the C3B will bind to this substance. Like I said, it can be virus, can be fungus, anything. It will bind onto this substance. But usually, this spontaneous C3B that is produced is unable to bind to our cells because our cells contain sialic acid, which will inactivate this C3B. And also we have other protective mechanisms, which I'll be telling you about. But for the case of the pathogen, it lacks this sialic acid. And because of this, C3B will simply bind onto that antigen. So the binding of C3B on that surface of the foreign antigen will attract another protein, the so-called factor B. Okay? This factor B, which you see here, okay, so the factor B coming in here will be split by an enzyme called factor D. So factor D will split factor B into BA and BB. Like we said, normally those with an A will not bind. So the binding is done by B. So the BB will bind with C3B to form C3B BB. But this C3B BB is actually unstable. Okay? So because it is unstable, it has to be stabilized by propagandin. So the moment it is stabilized, then that is what forms our C3 activation complex for the alternate pathway. So here we see first the second difference between the alternate and the classical pathway. The first difference we said was in the way that activation occurs. The classical pathway requires immunoglobulin M or immunoglobulin G, whereas the alternative pathway does not require these immunoglobulins. Two, we are now seeing that the C3 convertase in the classical pathway is made of C3, C2B, 4B, whereas the C3 convertase in the alternative pathway is made of C3B, BB, and P. P is the propagandin factor. That's a big difference. Now, the moment we form this C3 activation complex, the rest of the steps are the same. In other words, we split C3 into C3A and C3B. And then this newly formed C3B, we recruit more of the B, the D, and the P. And then we have the C3B, BB, C3B, which will give us the C5 convertase. That's another difference now. In the alternative pathway, we saw that the C5 convertase was composed of C4B to B, and then we added on the B, which came in from C3B. And then here we are saying that we have C3B, BB, 3B. Then after that, now this C5 convertase will cleave C5 into C5A and C5B. C5B will then start the process that will end up causing production of a membrane attack complex, the same way that we saw with the classical pathway. Okay? What about the lectin pathway? Now, the lectin pathway has properties that are similar to both the alternate pathway and the classical pathway. How? If you look at its activation, so the activation is through binding of the Mannan binding lectin. So this Mannan binding lectin will bind to mannose residues on the pathogen surface. So on these pathogens, the pathogen can be a fungus. So the pathogen itself has these mannose residues. So the Mannan binding lectin will bind on these residues, and in so doing, activates Mannan binding lectin associated serine proteases. These serine proteases are usually two. We have MASP1 and MASP2. So MASP1 is similar to C1R, and MASP2 is similar to C1S, meaning that these are serine proteases. These will then cleave C2 into C2A and C2B, and C4 into C4A and C4B, the same way we saw with the classical pathway. So here we see that this lectin pathway is activated by an antigen, but the first step of activation requires a serine protease, the same way that we've seen with the classical pathway. So the C4B2B will combine to form C3, and this will activate lectin pathway, the same way that we saw with the classical pathway. Now, the complement system has a potential to be extremely dangerous to the host tissue. So you can imagine that when you produce all these complement systems, you're producing cytokines. I'll give you an example of a disease that has killed people through cytokines. One of them is COVID. People who die of COVID are not killed by the virus, but they are killed because of the excessive production of cytokines, which overwhelm the immune system, okay? And they also destroy the tissues. So that is how damaging this complement system can be if it's not regulated. So because of this, activation must be tightly regulated through complement control proteins, okay? So these complement control proteins are in fact in much higher concentrations than even the complement proteins themselves. So we have more complement control proteins than the complement in the body. Reason being that complement has a potential to destroy the body, okay? Now, some of these complement control proteins are found on membranes of cells, okay? An example of this is the decay accelerating factor, also called DAF, which is found on the red blood cells. Now, deficiency of this decay activating factor means that this individual who lacks this DAF will likely get hemolysis. And usually this occurs at night. And because of this, the condition is called paroxysmal nocturnal hemoglobinuria. Someone wakes up and they are passing urine in the morning, which is bloody. Okay, that tells you that they lack the decay accelerating factor. Then 2 is that the classical pathway itself is inhibited by a protein called the C1 inhibitor. Remember, we said that C1 is very, very important because it is responsible for the downstream cleavage of R and S. Okay, so this one inhibitor binds and covers the C1 receptor so that Q doesn't bind. If Q doesn't bind, R doesn't get activated and S doesn't activated. And if R and S are not activated, it means that C2 will not be cleaved and also C4 will not be cleaved. Now, there are individuals who lack this C1 inhibitor. And like I will be telling you soon, these people normally come in with the edema that is usually around the face. Okay, so they end up with something called in familial edema. Then we have the alternate pathway. This one, its activation is normally limited by presence of endogenous complement regulatory proteins. So these are the so-called clusters of differentiation that are found on the self cell. Okay, so these are CD35, CD46, CD55, and CD59. Interestingly, pathogens lack these clusters. And because they lack these clusters, complement is able to bind onto the pathogens and not onto our own cells. An example is normally given. Protectin. This inhibits C9 polymerization. And yet we say that it is C9 polymerization which ends up forming the membrane attack complex. So our alternate complement system is able to recognize that these cells are self because they are presenting with these clusters of differentiation. Number 3 is that our cells, our host cells, are unable to accumulate C3b. Okay, because we said if you look at both the alternates and the classical pathway, the reason as to why cells get opsonized is because C3b tends to accumulate on the surface of a cell, and this increases its opsonability. But at the same time, the excess C3b will bind on either C2b4b to form the C5 convertase, or it will bind on the C3b-BBP to form the C5 convertase. So host cells are unable to accumulate C3b on their surface. And this helps to prevent destruction of these host cells. But also, in the alternative pathway, the host cells, the C3b that is generated during all these processes is usually rapidly inactivated. And this inactivation is done by factor H and factor I. Okay, so this factor H and I rapidly inactivate the C3b because we said that usually we have spontaneous hydrolysis of C3 into C3b and C3a. And in the presence of an antigen, C3b will bind. If there's no antigen, C3b will be inactivated. And this is done by factor H and factor I. In contrast, if you have an antigen, the internal hydroxyl group of that antigen will instead stabilize the C3b antigen complex, and that will activate alternate pathway. So that is the control, okay? So we have the C1 inhibitors, we have the decay activating factors, we have factor H, factor I inhibitors, we have clusters of differentiation of these complement support proteins that are usually found on the cells that are self and not on the antigens. What about the functions? What exactly does this complement do in the body? One is that complement is very important in formation of opsonins. The opsonins are C3b and C5b, and these enhance phagocytosis. Remember, we said that one of the major functioning of the innate system is through production of phagocytosis. It's very, very critical that we are able to phagocytose any antigen, okay? Because it is through this phagocytosis that we're able to produce peptides that are then presented before we can even start to make antibodies. So opsonization is very important. Two is that through opsonin, through complement, we're able to produce chemotaxins. So these are C3A and C5A, and these attract macrophages and neutrophils. And by attracting macrophages and neutrophils, you're literally starting a war, okay, and eliminating the organism. Then three is the production of anaphytoxins. This is usually the C5A, and these anaphytoxins end up causing degranulation of mast cells. That is why we know that when you activate complement, you might end up inducing an allergic response because you're activating degranulation of mast cells. Four is complement is important in production of membrane complex, which causes cell lysis. And through this cell lysis, you end up rupturing the membranes of the foreign cells. And then finally, this complement is important in agglutination. All these complement proteins that we've seen, right from C2A, C2B, C3A, C3, all of these enhance antibody production because they act as a unit, okay, to enhance antibody production, and this will enhance agglutination. Okay, so this is what I've been laboring to tell you about. So what about deficiencies? What happens if someone has a deficiency of a complement protein? The commonest really is usually deficiency of complement factors C5 to C8, meaning that someone is unable to form a membrane attack complex. These individuals are particularly at risk of getting Neisseria. Okay, so this, if it is Neisseria gonorrhea, someone will have recurrent gonorrhea, okay, simply because they are not able to form a membrane attack complex that destroys Neisseria gonorrhea, but also Neisseria meningitidis. Two, individuals who have severe liver disease, like we've said before, most of the complement proteins come from the liver. So if someone has severe liver disease, it means that they are unable to produce complement. These people will usually present with pyrogenic bacteria because, as we've seen, you are no longer able to produce opsonins, you can't produce anaphytoxins, you can't produce chemotaxins. So you end up coming down with pyrogenic bacteria. And then there are those individuals who have immune complex disorders. These immune complex disorders include SLE, patients who have rheumatoid arthritis. So these disorders will regularly utilize complements because there's a spontaneous utilization. And because of this, these people will have a complement deficiency. But at the same time, overactivation of complement means that this person will always have activation of a neutrophil functioning. So if it is rheumatoid arthritis, they'll come in with very painful joints, if it is the SLE, the skin will be damaged, the eyes are damaged, the joints are damaged, the heart is damaged. And of course, in the end, if you do an assay of complement, the complement will be reduced. Then there are those people who lack the C1 inhibitor, okay? These people will end up getting something called angioedema because they are unable to stop the production of C3A. So remember we said that C3A and C5A are anaphylaxins. So if you lack C1 inhibitor, it means that you're going to end up activating breakdown of C3A and C5. So if you break down C3, you end up getting C3A. You break down C5, you have C5A, and these are anaphylaxins. Because they are anaphylaxins, but also because they're going to increase histamine, histamine will increase vasodilatation, leakage of vessels, and then you end up with edema. So usually these patients will come in with familial angioedema. We normally manage this with a drug called methyropone. Then we have patients who have a deficiency of the decay activating factor or the decay accelerating factor. Remember we said that on the red blood cells, we have this decay accelerating factor which stops C3b from attaching onto these red blood cells. So individuals who lack this decay accelerating factor will end up getting complement-mediated lysis, and also the disease is called paroxysmal nocturnal hemoglobinuria. These patients will usually present with the passing of dark urine early in the morning. Then there are people who have transfusion mismatches. Imagine if someone has blood group A and you give them blood group B, you're literally giving them a different antigen. So you're going to immediately activate complement, and you end up with these transfusion reactions. So that is all about complements. Now I am going to open up the chat so that we can discuss. Okay?
Thank you, Doctor.
You're welcome.
Yeah, I have a question about, you talked about cytokine storms in late stage COVID-19 cases. My question is that, are there any complement inhibitors that have been allocated for prescription in COVID-19 patients?
Actually, we do not have complement inhibitors, because remember that complement is simply helping the other systems, okay? That is why what you want to do with COVID-19 is to start preventing viral replication early in the disease. You understand? So if you prevent the replication of the virus early in the disease, then you have less antigen to activate complement. Okay? But some of the drugs that we give, for instance, the steroids like DEXA, their role is not necessarily to inhibit complement, but to reduce the effects of complement on cells. Remember we said that complement will recruit cells like neutrophils. Now, if you give someone a steroid, it stops neutrophils from being recruited, and that prevents the inflammation. Okay?
Excuse me, sir.
Yes, please.
Excuse me.
Yes, yes. You can go ahead and and ask your question, please. You wanted
Yeah,
to?
this is Godfrey. Okay. I wanted to like inquire, like, do all the pathways occur like concurrently, or at certain infection, like certain, like one of the pathways is involved, or something, you know, like?
Okay, that's a good question. So remember we said that the classical pathway occurs when someone has had an antibody. In other words, you've ever been exposed and formed an antibody, right? So the first time you get exposed to an organism, you will most likely utilize the alternate pathway or the mannose binding lectin if you have a mannose receptor in the pathogen. But the next time you get exposed to the same antigen, all pathways will be activated. Okay? Hello? Hello?
We are hearing you, Doctor.
Yes. Hopefully I've answered this question. So the first time you get exposed, it is only the alternate that is utilized. Okay? Or if you have a mannose binding lectin, then also the lectin is utilized. But when your body has antibodies, then all three will be utilized. Okay, any other question?
Yes, doctor.
Yes, please. Please, can you ask? I can hear you. Okay, I haven't heard what you said, but I can check on the chat. Someone was asking whether bacteria develop resistance or virulence against these complement system mechanisms. So remember that complement is non-specific, okay? But also remember that we said that one of the ways bacteria can do is to actually get ways of overcoming complement. Either they'll cause lysis. If you remember Staph aureus, it has that capacity to stop your complement. If you remember Listeria, so yes, some of these bacteria can't. Then does that explain the amplified allergic response on subsequent exposure with the same allergen? The answer is yes, but this will become obvious when we study about hypersensitivity type 1. Okay? Hypersensitivity type 1 is the one for allergens. So the initial exposure is called a sensitization. And then after that, the subsequent exposures are more serious because of the cross-linking, which results into degranulation of mast cells. And then someone's asking, doctor, please recap on which Ig binds in acute and chronic. So if it's acute, it's going to be IgM. If it's chronic, it's going to be IgG. So do you have any other questions? Do we have questions?
I'm good. Good afternoon, doctor. I was inquiring on how membrane complex, the membrane attack complex works.
So the membrane attack complex is a cylindrical molecule. It's like a spear. Okay. So because it is like a spear, it literally pierces the cell membrane. And in so doing, everything within the cytoplasm pours out. Okay? Have you understood?
Hello, doctor.
Yes, please.
I'd like to understand how aspirin activates the immune system. How aspirin activates the system, the classical pathway.
No, you see, aspirin in some patients, they develop an allergic response. So it is not only aspirin, but all the other drugs. Okay. When you get an allergic response, it is the same. You're going to literally activate complement. It is an allergy. And this is worse actually in children more than adults. That's why they end up getting something called Raynaud's. Any other? So do you have any other questions? So I see Charles Lugaju. Charles, you can ask. Charles, unmute yourself and ask. Yes, Charles.
Yeah, thank you, Doctor. So my question is, in cases of deficiencies of complement,
yes,
are there any tests that are tailored to enable a health worker to be able to diagnose that?
Yes. We can do those tests which look for complement levels. There are tests that we do. Okay, so I see someone who calls herself Miss World. Yes, Miss World, you can ask.
Good afternoon, sir.
Good afternoon, how are you?
Fine, thank you for the lecture. It has really been beneficial. However, my network cut me off badly on the lectin pathway. I missed out on that. I don't know if you can do an overview, please, even if it's for 1 minute or two.
So the lectin pathway, the only difference is that you have manus binding lectins. So imagine a cell, okay? And this cell has manus receptors, okay? So the manus binding lectin will bind, and this will activate the manus associated serum proteases. So these are two proteases. There is MASUP1 and MASUP2. These act as R and S. So we saw the serum proteases R and S, which cleaves C4-2 and C4 and C2. Remember? So MASIP1 and MASIP2 act like C1R and C1S, which are serum proteases. So the moment you have these manam-associated serum proteases being activated, then they will simply cleave C4 and C2 to give C2A and C2B. And then the rest is like what we saw with the classical pathway. C2 will bind with C4 to form C2, 4B, C2B, 4B, which is your C3 convertase. Okay. So Miss Ward again. Hello?
Yeah, I've gotten you. Thank you, sir.
You're welcome.
Another question, sir.
Yes.
Won't the C3A and C5B, if they're over, can I say, if they're in excess, won't they cause over or hyperreactivity?
Which ones?
The C3A and C5A.
So the C3A and the C5A,
Yes.
obviously, the body has its own limits. Okay, and that is why if you have individuals who have severe allergies, the
Yes.
next time when you get, so each allergy is normally an increase of the previous allergy. So they tend to increase with each exposure. So yes, the more you break down C3 to produce C3A, and the more you break down C5 to produce C5A, you produce more of these anaphylaxins.
Okay. Okay, thank you, sir.
Actually, that's why in some of these cells, we simply give them mast cell stabilizers. So the mast cell stabilizers act on the membrane such that even in the presence of these other components, the mast cell does not degranulate.
Okay. So then which drug is given in patients with angioedema? I beg your pardon on that.
It's called metylapone, but don't mind, no one can ask you that. So what's important, again, to recap, is to know the 3 types of activation, the differences, the steps in activation of each of those, the role of complement, the 5 that we've talked about, and then the regulation of the classical pathway and the alternate pathway, and then the diseases that can occur if there is a deficiency of either the complement or the regulatory pathways. As simple as that. So if there are no other questions, then I think we are going to stop this lecture here. And I'm going to wish you, oh, there's someone called Eva. Eva, you can ask. Then there's God's love. Eva and God's love, you can ask. So we can start with God's love. It seems God's love is not ready. She's still praying. Let us go to Eva. Eve.
Good afternoon, sir.
Good afternoon, Eve.
I'm requesting to know why is it that for acute infections we have IgM and for chronic we have IgG. Same question. And another question is that why do we have, when someone has just gotten the infection, why do we have IgM very high and IgG very low? Yeah, I would like to know why.
So have you guys done immunoglobulins and immunoglobulin switching?
No.
Ah, okay, that's the reason. Okay, that will become obvious when you start about immunoglobulins and immunoglobulin switching. So when you get an infection, immunoglobulins have a heavy chain and a light chain. If you can remember some of the biochemistry you did, right? So the switching occurs at the heavy chain. Okay?
Yes.
And this heavy chain is switched based on the longevity. So this will become obvious when we do antibodies. Okay?
Mm. Oh, thank you very much.
You're welcome. Jacob Ochen. Jacob. Jacob Ochen.
Thank you, Doctor. Previously, I couldn't unmute. My question is,
yes,
these bacteria, haven't they evolved and maybe come up with any virulence against the complement system?
They have. They have. That's why you see, if you can go through, and of course, you're going to look at some of these things, mechanisms that bacteria use to overcome the immune system. I'm sure there should be a lecture on that. So one of those actually is an effect on the complement. Bacteria can stop your complement. The example I was giving you is on Staph aureus and all those intercellular bacteria. They're able to withstand our complement. Okay, any other question? So if there are no other questions, hello? If there are no other questions, this work is going to be available onto your LMS. Please go through and also get some textbooks and read. Actually, complement is one of the simplest things to understand because it is literally learning that the A's are usually light, so they diffuse away, but they're also very important in allergic reactions. The B's are normally heavy, so they combine, okay? And then it is simply knowing that you have 3 activation mechanisms. So we are going to stop here. Some of you, unfortunately, have very poor network. Okay. So I am going to stop this lecture. We shall meet again in the next lecture. Thank you and enjoy your weekend.